now we will be talking about the hallmark features of cancers. What are they? They are first of all self-sufficiency for growth factors, insensitivity to growth inhibitors, altered metabolism, evasion of apoptosis, immortality, sustained angiogenesis, invasion metastasis, and evasion of host-immune response. So these are the hallmarks of cancer. Now we'll be talking about the genetic basis of carcinogenesis. What is the genetic basis of carcinogenesis? In which we are firstly going to see that in a cell we have 4 types of genes. They are proto-oncogenes, tumor suppressor genes, DNA repair genes, and the regulation of apoptosis. The proto-oncogenes are the normal genes that are present in our body, and they act as an accelerator, and their role is that they perform increase in gain of functional mutation. Then if we talk about tumor suppressor genes, they act as brakes, and they are responsible for the loss of functional mutation. Then if there is also a mutation in the case of DNA repair, it can also cause a cancer. And similarly, if in the case of regulation of apoptosis, there is a mutation, then it also can cause cancer. These are the 4 genes by which any mutation in them can lead to a cancer. Now, first of all, we will be talking about the self-sufficiency in growth signals. It means that the cancer cells are self-sufficient in having the growth factors responsible for their growth, so they don't need growth factors from outside for the same. Okay, like if we talk about the proto-oncogenes, they'll produce oncogenes. Normally in our body, proto-oncogenes are present, right? But when there is some mutation in a body, then the proto-oncogenes get converted into the oncogenes. Oncogenes are cancerous genes. Why? Because they produce oncoproteins which are responsible for causing cancer. Okay, so now if I talk about a normal overview of the cell, then what happens? There are 5 factors, there are 5 levels at which we can study this. So first of all, when the growth factors comes at the growth factor receptor, it will form the signal transduction protein that will form the nuclear transcription factor, and that will form cyclins. And cyclins are finally responsible for the proliferation of cells, and when there is more replication of cells or more proliferation of cells, then it will lead to cancer. Right? So what are the points that are associated with it? It can be due to point mutation, can be due to overexpression, can be due to translocation, whatever. Okay, so there would be alternation in the activity of certain gene cycles that will lead to the proliferation of cells and finally into the cancer it will cause. So we are going to study it at the 5 levels that are the growth factor regulator, the signal transduction proteins, the nuclear transcription factor, and your cyclins. So talking about the growth factors, first of all, there are 2 types of genes. There are 2 types of growth factors. One is your platelet derived growth factors, that is regulated by cis gene. More, that is increase in the amount of cis gene will lead to glioma. Then there is hepatocyte growth factor. It will lead to hepatocellular carcinoma. The increase in the HGF gene will lead to hepatocellular carcinoma or the liver cancer. This was all about a growth factor. Talking about the growth factor receptor, it is the receptors that are associated with tyrosine kinase activity. We can say it as an epidermal growth factor, which is more the tyrosine kinase activity. So there are 3 types of genes we're going to talk about here. That is your ERBB1 gene, ERBB2 gene, and the RET gene. Talking about the ERBB1 gene, this gene is involved, and it would be associated with lung cancer. ERBB2 gene or the HER2 gene is associated with your breast cancer, and your RET gene is associated with your MEN2 syndrome. What is MEN? MEN means multiple endocrine neoplasia. This is your RET gene. So it will be associated with your pheochromocytoma, medullary carcinoma of thyroid, right? So this was all about your growth factor receptors. Now thirdly, we are going to talk about the signal transduction proteins. So in this signal transduction proteins, we are going to talk about some particular genes, that are the RAS gene, the ABL gene, the BRAF gene, and the beta-catenin gene, right? Talking about the RAS gene, it is the most commonest type of mutation, and you can see in the cell what happens. There is a GDP RAS present in the inactive. This is the inactive state of the GDP, that is GDP RAS. When due to the growth factors it gets converted into the GDP RAS, there is increase in the amount of GDP RAS, and this is the phosphorylation that is taken place. Now there will be further, like AKT gene will increase, MAPK will increase, that will lead to cell proliferation and the replication of it. So actually what has happened, from from an inactivated state, that was your GDP-RAS, that was the phosphorylated state was formed. Now it has lead to the production of factors which has increased the cell proliferation. Now it can also be reversible, like due to the due to GDP phase and the formation of neurofibrinogen, it will lead to again formation of GDP-RAS from GDP-RAS, right? This was the mechanism of your RAS gene that was involved in it. Now the subtypes of it are like the KRAS gene is responsible for colon cancer, HRAS gene is responsible for your kidney and your bladder cancer, and NRAS gene is responsible for melanoma. Talking about the ABL gene, it is a non-receptor tyrosine kinase activity. It shows it is due to the process of translocation that takes place over here. Like there is ABL gene present in the chromosome number 9, BCR gene present in the chromosome number 22. So due to the translocation of 922 chromosome, there is a formation of fusion gene, that is ABL BCR gene. ABL BCR, they are present on the single chromosome due to the process of translocation. Now that will lead to an increase in the tyrosine kinase activity and will stimulate the proliferation of cells, and finally cancer will take place. Like this is the D922 chromosome is called as your Philadelphia chromosome, present in the case of CLL or ALL, right? इसमें क्या होगा? Oncogene, there would be presence of oncogene addiction in this. What is oncogene addiction? When tumor cells are so much dependent or addicted to tyrosine kinase activity, means then and only the tyrosine kinase activity increases. There is oncogenic formation, cancer cell formation, or the tumor cell formation. If there is no increase in the activity of tyrosine kinase, there it won't take place. So we have to act on this target to decrease the tyrosine kinase activity so that the oncogenic addiction is broken. So this is how we can treat this type of cancers. There is a targeted therapy that we give through the imatinib drug. It is this ABL gene is more effective against the CML than ALL. I mean this imatinib drug is more effective against CML than ALL. Right? I'm talking about the BRAF gene. BRAF gene is actually responsible for the stimulation of MAP kinase that leads to cell proliferation, and it is seen in the cases of hairy cell leukemia. Means 100% of the hairy cell leukemia means it is strongly associated with your BRAF gene or the increase in the MAP kinase activity. Then your benign nervous, that is 80%, and melanoma, that is 60%. So we have discussed 3 genes, and the 4th one is beta-catenin. Beta-catenin leads to the increase in the MYC activity, MYC gene activity that leads to the increase in the cell proliferation. There are some tumor suppressor genes that are responsible for controlling catenin or inhibiting catenin, that are APC gene and your E-cadherin. An example of this is colon cancer. So under the topic signal transduction proteins, we discussed 4 genes over here. That was your RAS gene, ABL gene, BRAF gene, and the beta-catenin gene. Now let's talk about the nuclear transcription factors. MYC gene is an example of nuclear transcription factor, and it is a master regulator of cell proliferation. This is the master regulator of cell proliferation, and it is responsible for increasing in the cycling activity. It's responsible for increasing the telomerase activity so that there is not shortening of the telomerase and thus replication will not stops and the cell replicates on further. It also is responsible for reprogram, that is also called as your stemness. Now what are the further subtypes of MYC gene? That is CMYC, responsible for your Burkitt's lymphoma. Burkitt's lymphoma is having the maximum mitotic activity and the fastest rate of proliferation. Now talking about the NMYC gene, it's responsible for neuroblastoma, and LMYC is responsible for your lung cancer, that is your small cell lung cancer. So C for Burkitt's lymphoma, N for neuroblastoma, and L for lung cancer, right? Now talking about the 5th factor, that is your cyclins. So if we talk about cyclins, they are having an intermittent activity, that is they can be like in switch like an on-off mode, and they are responsible for this on-off and intermittent activity cycle responsible for production of CDKs, that is a cyclin-dependent kinases that leads to phosphorylation. Now there is a particular sequential activation of cyclins, and what are this? First of all, cyclin D is responsible for the activation of the CDKs 4 and 6, E CDK2, cyclin A CDK2, and cyclin B CDK1. And how can you remember this D, E, A, B cyclins that are in the sequential activation? step, that is dhoni ek akela batsman, that is DEAB 46221. Fine. Talking about a particular cell cycle now. So there are 4 phases in a cell cycle: G1, S, G2 and M. When you come from quiescent phase to G1 phase, then from G1 to S, there is a checkpoint present in between G1 and S, and the second checkpoint that is present between your G2 and M. If we talk about the checkpoint present between G1 and S, it is regulated. The first step is regulated by your cyclin D or CDK4/6, that is associated with mantle cell lymphoma also. So this is very important. At the initial step, that is from the G1 to S, initially it is regulated by your cyclin D or the CDK4/6. Then from coming towards the S phase, it was cyclin E or the CDK2. Then from going to S phase to G2 phase, it is cyclin A, that is CDK2/1. From G2 to M, it is cyclin B or the CDK1. So what are they in the sequence? Cyclin D, E, A, B. Fine. And so in the G1 phase or the S phase, the gene that is responsible is your RB gene or the P53 gene. And in the G2 or the S phase, the responsible gene is the P53 gene, and that's why the P53 gene is also called as the guardian of genome. The P53 gene plays role in both the checkpoints but greater activity in the checkpoint number 1. If there is an overactivity of the CDK4 gene, then it will lead to sarcoma and brain and gallbladder cancers. If there is an overactivity of cyclin D1, then it will lead to mantle cell lymphoma. Okay, so now if I talk about a CDK activity, that is cyclin dependent kinases activity, you can increase their activity by cyclins and increase their activity by CDK inhibitors. So now we'll be talking about the CDK inhibitors. There are 2 types, non-specific and specific. Non-specific are P21, P27 and P57. As the non-specific, so they inhibit all the CDKs. But if I talk about the specific CDK inhibitors, they are P15, P16, P18 and P19, right? They inhibit only CDKs 4 and 6. Like cyclin D is important for proliferation inhibition. So this was all about your first of all we talked about the genetic basis of carcinogenesis, the self-sufficiency in growth signals. Thank you.